It's 6:30 p.m. right now. So hi, thank you once again for joining us tonight in this session. So allow me to do a rough introduction for myself. Hi, I'm Ming Xue here. You may have noticed my email that I have sent to everybody. I'm a representative, a marketing staff from PSB Academy Private Limited, School of Life and Physical Sciences here today. So I think what we are doing right now, it's an ongoing series that we have been trying to advocate for PSB Academy, which is to always educate the public. And as such, right, we are also conducting a series of online talks and trial classes. So today you're joining us in our life science trial classes, headed, of course, by Dr. Seah, as you can see in the screen over here. So Dr. Seah is also our resident lecturer with PSB Academy. So if any one of you were to sign up for a apply for a life science degree program with La Trobe University in partnership with PSB Academy, you'll most likely get Dr. Seah here as your lecturer here, because he covers almost all the life science topics that we have here. And Dr. Seah has been with us since for the last 4 years as well, and he's our module leader. He's always constantly innovating topics for us to really let everybody improve. Just another little brief introduction. The platform they're using right now, it's also known as the Blackboard platform. So with COVID-19 situation, most of the sessions are done online. So likewise, this is a session that is also our actual university students. They always use the Blackboard as well to attend lessons. Of course, these are not forgetting inside your coursework, there will be such thing as lab work as well. So without further ado, I'm not going to hear me blab so much about what's PSB about. I think you guys are here for a brief lesson. I'll pass the time on to Dr. Seah, and we can do a roundup later after that. All right, Dr. Seah, over to you.
Okay, so thank you, Ming Xue. So welcome all the participants and potential science students. So what we're doing here will be to share with you some insights about how university education is conducted. So I'll be doing a trial lecture. It won't be a full lecture series, but it will be just a glimpse of about 45 minutes to 1 hour, where you will get to understand how concept starts to build up and relate to the bigger picture. So when we look at neurocommunications, which is the theme for today, we are more interested to understand not just how brain works, which is basically at the systemic level, but are we able to zoom in on the details to understand, for instance, how processes such as neurotransmission is being carried out, and they have to be carried out in an efficient manner so that we are able to then allow the effective neurocommunications between different parts of the brain and also different parts of the body. So to reduce the bandwidth and the lag, I'm going to turn off the video, and I'm going to show the PowerPoint slides. And please allow me to bring you through 45 minutes of a learning journey, trying to understand a little bit about molecular details of how neurotransmission is basically achieved in the human body. So let's get started with the slides. So for the first slide, it will be just an introductory trial class that we aim as a continuous set of programs, particularly to give the public some of the information that is available, as well as to introduce some aspects and nuggets of science so that we are having a better understanding of it. So today, we will have a short talk, particularly on neurocommunication. So our focus today is on how the brain is able to carry out its function. So particularly when we look at the term neurophysiology, neuro, if you think of it as the root word neuron, will be linked to a cell from the brain. And we are more interested in understanding the physiological aspects, such as when we look at it, how the neurons is able to achieve its normal role and function. And what is its normal role and function, you may ask? It is to carry out efficient signaling between itself, between parts of the brain, and definitely across different tissues wherever is required. So for simply, we can actually say that neurophysiology will then encompass the idea of how a neuron is able to fine-tune its signaling to allow the effect, particularly on the target organs, to be achieved, including controlling of muscles, for instance. With that, we are going to just briefly talk about 3 major learning objectives, and at the university level, each lecture will have clearly defined learning objectives, which will then shape the direction of how the knowledge will be imparted. And subsequently in the slides, we are going to talk about, firstly, identifying the source of an action potential. And in this particular aspect, we are going to look at how it all begins. How an action potential eventually can be a wave of impulse and signal that can be propagated to the effector tissues, effector organs. And as we bring it back into the big picture, we want to establish a comparison against the greater potential, a progressive effect versus a local effect. And finally, we will then try to sum up and provide perspective in terms of how a nerve impulse is being initiated. What are the factors that will then drive the efficient propagation of the nerve impulse so that it can reach its intended target organ? So it can be, for instance, an impulse starting from a portion of the brain that will then carry its long journey, for instance, to then trigger a tissue-related effect, including, for instance, contractility of muscle. So if you look at the diagram that we have, the brain is a very complex organ. It is responsible for a lot of events, and some of those are listed on the diagram. Reflex event, posture for maintenance, emotion, anxiety, as well as basically action. So given that the brain is so critical, we may ask ourselves, how is basically this systemic effect of the brain in carrying out neurotransmission being achieved at the cell level, at the molecular level? So in this particular slide, we are showcasing the different aspects of a synaptic connection. The neurons is not going to stand alone. It is going to interact with other neurons like itself, and it may have to, at times, be able to extend the signal, integrate the signal, so that we can then pass on the signal to achieve a physiological effect that is intended for the tissue, which are the effector targets. And in saying so, we want to understand how these parts are able to work efficiently, how signals is being propagated. So across the lecture, you'll find that we will talk about the cell organization and particularly about neurotransmission, which will be then moving on, particularly from the axonal hillock all the way down this long axonal body, the axonal cell length, until we are able to connect to the next cell at the synapse. So with that, we have a next picture that basically highlights the 2 major systems that is found in the body. The idea here is not for us to go into details, but is to allow us to appreciate that all the different organs, tissues, are well connected back to the brain, directly or indirectly, and they are all subjected to nervous control. Nervous control can then take 2 forms. One of them is sympathetic nervous system, which will most likely control the voluntary as well as the involuntary aspects in conjunction with the parasympathetic nervous system. So the idea that we want to highlight is the nervous system are able to coordinate all the major activities that we see in a human body, and this will then govern, for instance, events which are important for homeostatic control, including hormonal secretion. So just to recall about what we typically will cover as a university style of lectures, we will then recap some of the points that is covered in the previous class and then build on and extend the concept. So this slide basically highlights some of the aspects that will then support the understanding of physiology. So a few things that will be critical to allow a cell to achieve efficient neurotransmission. The first one will be the uneven distribution. And when we're looking at this particular uneven distribution, we are referring to ions. Typically, the two major positively charged ions, sodium, which is denoted by the symbol Na, and potassium, which is denoted by the symbol K. You will find that typically by normal consensus that we are able to obtain from experiments, sodium exists at a higher concentration outside the immediate cell environment. We term it as the extracellular fluid, while there will be a smaller concentration of sodium within the cell. So likewise, this is basically denoted by the size of the element that is indicated. On the contrary, we'll find that there is a preferential accumulation of potassium within the cell. You may be thinking at this point, yes, it is differentially distributed. There appears to be a higher concentration at one side compared to the other, as if there are compartmentalization differences between the exterior and the interior of a cell. But when we look at it, this is achieved because the cell has an intact semi-permeable membrane. So the next question we may ask will be, why and how is it achieved? Inside the cell, a specific motor, which is the sodium potassium pump, or ATPase, which is an enzyme that will require the use of ATP hydrolysis to provide the energy to differentially distribute the ions. In other words, the operation of this pump generates a difference where there will be a preferential push of sodium outside the cell, and this will lead to a concentration that is higher for sodium outside, whereas potassium is basically higher inside the cell. So the continuous operation of this pump generates that difference, which we term as an electronic gradient. So what is the impact of this? And is there only positive ions? We do have also negative charge ions. Chloride is one example, Cl minus. So when we integrate the understanding, as what we mentioned from earlier classes where information is built on, we will have to look into the membrane properties, including whether this membrane may indicate a specific level of leakage. And typical leakage that we can see includes potassium leakage, chloride leakage, and how this basically integrates into the big picture of how the membrane potential is being specified by the combined effect of all the ions, the concentration, and because these ions are charged, the direct movement of ions inside and outside a cell do bring about a change in the relative membrane potential, which we denote as RMP. So in summary, when we look at this relative membrane potential, we will have to look at the cell at the resting state. So RMP is also denoted as the resting membrane potential. At this instance, we are basically trying to highlight the uneven distribution triggered by the effect of a molecular pump brings about a difference in charges that will then causes a typical resting membrane potential to be negative. So after we have set the stage, the next part will then be looking at how this is being achieved. So let me just tweak it towards the aspect here. So I'm going to play a video. Okay, give me a while. Let me turn up the volume a bit. So
floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside the cell's interior,
okay, so let me just play again for missing out the front part.
There's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside the cell's interior.
Okay, so what we have basically attempt to illustrate is there is an existence of a difference. This difference, which is indicated by the resting membrane potential, is also brought about by the differential accumulation of ions, particularly potassium within the cell. So in other words, when we look at a cell, such as a neuronal cell with the cell body, as well as the difference in charges, we typically see the interior of the cell being more negative than the exterior. So this charge separation will then be able to help us to understand the electrostatic as well as the concentration gradient that together influence this equilibrium. So meaning that as we look at the resting membrane potential, you may be asking, is this a fixed value? Is it subjected to change? Yes, indeed. It is subjected to change whenever there is a stimulus. And you may be then wondering, how do we basically trigger the change? The change can be triggered by differential movement of ions whenever permeability is facilitated. So at this junction, when we look at how this is being carried out, we will have to look at the key properties of this sodium-potassium pump, which is the ATPase that we have mentioned. So earlier we mentioned that, yes, it preferentially sends the sodium outside the cell, but it also brings about a net intake of potassium inside the cell. So how does it work? So I'm going to play a quick snippet to actually highlight this point.
The membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium and a difference in charges, making it more.
Okay, so what we have seen in that short clip is the membrane is having a positive and a negative difference by virtue of the action of the pump. So in terms of how the pump operates, there will be a 3 sodium versus a 2 potassium change. And hence, as I lose 3 potassium, as I lose sodium outside the cell, I'm able to compensate with 2 potassium as the positive charge. In other words, I am losing 1 net positive charge every time this pump actually operates. The continuous operation of this pump will then bring about a relative positive exterior with a relative negative interior in terms of the membrane potential change inside the cell. So at this junction, we are going to look at the channels that allows these ions to move.
Channels which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter like serotonin or a hormone latches onto it. And we also have mechanically gated channels which open in response to physically stretching.
So based on that short clip, we see that channels are important. They are acting as gatekeepers on our membrane. Earlier, we mentioned that the membrane is subjected to permeability changes, and that can be attributed basically towards the frequency and the chance whether these channels are going to open or not. In other words, if these channels are operating and they are allowing the passage of ions, we will most likely see a redistribution of these ions. At this junction, when we come back to the main picture, we'll find that the membrane, which is semi-permeable, there is some form of permeability that is associated, and every time the channels are going to open, and some of the channels that are critical will be the voltage-gated channels. What it will do in this case is, if we are considering the directionality, and since the potassium and the sodium are differentially distributed, the moment we have the voltage-gated sodium channels being open, we will most likely see that sodium will go into the cell. What this actually triggers is it creates the start of an excitation event. So in other words, this excitation will actually lead to a corresponding changes, particularly in raising the membrane potential. So in simple terms, what it's trying to say is ions start to rush in whenever the voltage-gated channels open. The net effect or the consequence will be we are going to see this as a positive and a reinforcing effect, typically feeding back to increase the membrane potential from a negative value towards a positive value. So this particular event, the switch of a negative membrane potential value towards a positive membrane potential value, is termed as depolarization, an event that makes the cell excitable. Correspondingly, when we look at the potassium, potassium has the tendency to actually exit the cell. And every time when we are able to achieve the opening of the voltage-gated potassium channel, which are typically closed and they open in a sequential event, only after the prior event of sodium-induced excitation has been carried out. So this is, in other words, a delayed event. The effects that it causes will be, it will lead to a negative feedback, and typically it leads to an opposing role where, unlike that of sodium, sodium causes the membrane potential change from a negative to a positive value. Here, the movement of potassium will then trigger a change in membrane potential from a positive value towards a negative value. But there is a tendency, particularly for the potassium channel, to overcorrect itself. It may at times go into not just repolarization mode, but it may overshoot into the hyperpolarization rim. And correction will be required, and that is the typical job of a sodium potassium pump. So these are the changes that we see that is happening at the cell that triggers the change from depolarization to repolarization and a potential overshoot leading to the hyperpolarization in the negative rim. So likewise, in the positive rim, there is also a chance for overshoot, but typically because of the progressive and sequential opening of the potassium channel, this is often not being demonstrated by a cell. So in other words, as we zoom in on the key role, we'll find that the membrane potential tends to go from negative towards positive, tends to come down, and if it needs, it will then be basically restored back to the resting membrane potential once again. So far, what we have actually indicated is changes in levels going up or going down is due to the permeability associated with ion channels, And because of the passage of ions, and ions carry charges, this electrogenic change will then make the cell develop and achieve two phases. One, excitation, which is coupled with depolarization event, and subsequently a repolarization, which is basically going to help you to restore back to the normal non-excited status or the resting status. So when we take a closer look at how this voltage-gated channel is going to achieve the role, we are going to look at the charges that is distributed. And in fact, this voltage sensor is highly sensitive towards the type of ions that is supporting. So in other words, when we look at a sodium ion channel, it is only able to permit the entry and the movement or the passage of sodium and nothing else. They are highly specific. This specificity is basically governed by the presence of amino acids. They are typically positively charged, which you can see in this pink arrow and this pink column with the positive charge amino acids. And this basically causes that restriction for the ion species. So now, since it is going to be so specific, when we look at the effect, particularly at the electrogenic level, changes in membrane potential, we are going to then look at the mechanics of how they open, how they close, and of course, what's important is the concept of time. So when we look at the first type, which is indicated for the voltage-gated sodium channel, there are 3 states. For the potassium channel, there are 2 states. The difference between them will be the voltage-gated sodium channel is able to open and close in a rather rapid manner. And because we've mentioned that the potassium channels will open sequentially after the sodium channel has closed, the net effect that we are going to see will be at the cell level, a depolarization coupled with increase in membrane potential coupled with a subsequent drop leading to hyper or repolarization. So this will be just a quick graphical representation of the 3 states. Ideally, you may be thinking, yes, it opens and closes for 2 types. What is so special? How is it able to achieve a rapid inactivation? So what you see here is there is a ligand. In other words, there is this domain that is movable, that is flexible. It is capable of triggering a direct blockage. In other words, it acts as an inactivation mechanism. In a simple analogy, you may think of it, if you are watching a show, you buy a ticket and you are about to enter a cinema, the tickets are allowing you to enter as a passage. But suddenly, if I need to stop it, I can get a security guard to actually block the faggots. And this provides a means of rapid inactivation as a means of regulation. So simply means I can block it instantly by getting someone to just block it, whether it's a big fat security guard blocking the door, or whether I have an inactivation device such as the ligand. So when we compare them in terms of the rapid inactivation, you'll find that it's going to occur only after all the sodium channels have been opened, but you want basically an immediate closure. The immediate closure will give rise to a subsequent delayed opening of the sodium channel. So what we have seen so far is to excite a cell, you need to bring the membrane potential up, and that is basically due to the fact that the sodium channels are opening. And subsequently, to achieve the effect of bringing the membrane potential down, which is much more slower, and the fact that these potassium channels are sequential, we will relate them into the big picture looking at conductance and how this conductance is then allowing us to interpret changes, not just locally, but also ability to spread the signal. But before we revisit that, we will have to think of situation should there be no voltage-gated channel. What may happen? Now, the situation will be as such. Depending on the intensity, the stimulus may spread to the neighboring patches of the membrane, and that will constitute an area that is directly imposed as the area of depolarization. In simple terms, what it's trying to say is if I have a signal or stimulus such as, let's say, a pen, you poke your skin, it will have the effects felt in the nearby patches of the skin. But eventually it will dissipate off because the signal will sort of even out. So in other words, to have the signal being capable of propagating, the stimulus has to be strong, has to be reinforced. So when we revisit the fact that sodium channels, when they open, we will find that the potential increases, and it is a positive reinforcement feedback mechanism. So when we bring all these context in the big picture, we will be able to understand. So it is important for us to have a good, strong stimulus, and it is important for us to have all the ion channels that is going to open so that all the ions can move and cause the change that is required. So is that all? But it's not all yet. So when we look at the nerve impulse and its generation, yes, when we have a stimulus that is activated, it may go sideways and potentially being dissipated. The question of how we can reinforce the signal so that it carries on its signaling and transmission is the concept of depolarization. Decremental spread, particularly, that triggers an earlier event can be reinforced. And this reinforcement triggers a progressive wave of depolarization. Think of it just like the seaside, the ocean. When the waves reach the shore, the next set of waves come on and come on in a repetitive manner. So how do we achieve that? And that will be important when we look at the big picture, how to achieve neurotransmission in a highly organized and efficient manner. So if you take a look at the animation, the charges basically is going to be dissipated. Signals are going to be moving down the axonal body. So signal is going from the left to the right side of the panel. So to trigger that, a progressive reinforcement, positive feedback, and continuous release to sustain this signaling is essential. So when we look at the big picture, how do scientists actually study it? Back then, in 1988, more than 30 years ago, how do they study that? They put tiny electrodes into the neurons, and with that, what they do is they try to measure the potential changes and then correlate it to different parts of the neuron. So in other words, from such an early study, and then they revisit the concepts, we are able to understand exactly, based on all these microelectrodes, which parts of the neuron are sensitive to membrane potential changes, and should we have the ability to reinforce them, that part or that region will be important to initiate the action potential. So what this study found is there is a region that is termed as the trigger zone. The next question that you may be thinking, why is it so special? Why is this trigger zone so special? It is special because it has the highest density of these voltage-gated sodium channels, or even the potassium channel. And by virtue of a high concentration of these channels, they allow for efficient propagation of signals. But when we think of it back into the context of a nervous impulse, as a singular nervous impulse, I typically will have to activate before retrieving the signal back until it reaches the normal resting membrane potential. So is there any mechanism that is available to prevent any overexcitation? Because if you have overexcitation, You may then suffer from some disorders such as fits and worst case epilepsy, as well as other form of muscle twitches and so on. So a built-in mechanism particularly linked towards the absolute refractory period, which is pinpointed as a phase which is marked as red. Further excitation is not possible. As long as all the sodium channels are open and before they are totally closed, technically there is no way for you to excite a cell. As long as it is falling within the first 1 millisecond upon the initiation of the initial action potential. So this is like a fail-safe mechanism. Of course, if you think of it at a bigger picture, it is to prevent overexcitation, which may then tire the cell, and then the neurons may die. So likewise, if you are looking at the region that is marked as the yellow region, termed as the relative refractory period, there is still the chance that you can excite the cell, but not at the absolute refractory period. Think of it, it's just like a circuit breaker to prevent overexcitation. Coming back into context, when we look at the relative refractory period as well as the absolute refractory period, the concentration of the ions, particularly for the ion channels, as well as the relative of closure will be critical. So why is it that we mentioned that this particular region is going to be the circuit breaker? It is because of the fact that the sodium channels are rapidly inactivated. They are not going to be active anymore. There is no way, in other words, at this point in time, due to the rapid inactivation of the sodium channel, to kickstart another action potential. This means you cannot progressively trigger action potential without a recovery phase. The recovery phase will basically take easily 3 milliseconds. So when it reaches this phase, back to the resting membrane potential, you can excite again. And even if you are going to excite, let's say at the relative refractory period, you will still require a larger than normal impulse to be able to trigger that, because you are going to see a slow progressive recovery of the sodium channels, which is going to become open progressively, but the potassium channels remain closed, and hence a higher stimulus is required to effect the same level of membrane change. So at this junction, what we have seen is trying to relate cellular events that lead to ion changes, and these basically accumulates in transmission event of signal. So when we look at transmission event of signal as a broader picture, you'll find that yes, in the relative refractory period, the status of the channels being open and closed are being shown here. So this will eventually move from a negative state towards the activation domain, where we typically will move towards a positive value. So as the action potential propagates, what we are going to expect to see at the axonal heel lock, which is defined as the region termed as the trigger zone, is that the first event for depolarization is, as what we have recalled, sodium channels have to open. And when they open in large amount, that causes a membrane potential change, and if it achieves a positive value from an initial negative value, we are more than confident to say that there will be most likely an action potential being triggered, and that is your first cellular signal. So when we look at the absolute refractory period, just to recall, it is to prevent one wave of depolarization following the next wave. This rapid inactivation acts as the immediate circuit breaker to prevent that. So at this juncture, you may think of it as molecular events are critical to trigger the transmission of the signal. But when we think of it, the cell is smart enough to put in measures, to put in circuit breakers to prevent overexcitation. So this is a very elegant way. I control signaling, I control the magnitude, the duration, as well as the intensity, but I can control when to stop as well. So the cell provides this mechanism to prevent excitation in a very drastic state, which may lead to cell death. And as what we have known, progressively when cells such as the neuronal cells start to die, we may come up with neuronal degeneration disease. So in the aspect of how this signal is being conducted, you'll find that the signal is being propagated progressively. This is a very highly efficient manner during which there are going to be nodes, intermediate points. Think of it as a traffic junction, which will allow the fast and efficient movement of charges with two key criteria. One, I have myelin, which is actually an electrical insulation that is brought about in the system. And the effect that it is able to do so helps us to govern the properties of electrical insulation. Think of it, it prevents the loss of the current, and hence the magnitude of the signal can be a lossless one. I start off with 100 units of signal. Most likely I will receive near 100, if not 99 units of signal at the recipient end. So this is being reinforced with the progressive boosting and the regeneration of potential at each of these nodes. And this is critical, coupled with the electrical insulation, to reduce the loss of signal. So what we can see is that the cell, as well as the arrangement of neuronal conduction between cells to cells, neurons to neurons, or between different parts of the brain to the affected tissues, is going to work on the premise that conduction of these impulses has to be efficient, has to be lossless, and that is critical to ensure the function. So this will be just an animation. You'll find that changes in the charges will be as if like a wave, and there will be a transient depolarization change in events. And what is more important is, under this fatty myelin shift, the ions is able to move efficiently across. So the combined mechanisms allows us to be able to achieve neurocommunication in a very effective manner. So when we look back, as we first started the talk 20 minutes or so ago, we want to look at the big picture. We go zoom in on the small picture, and from the small picture, we are able to then see how cells, how proteins on the surface of cells acting as ion channels allow this marvelous transmission event to carry out flawlessly. So this is just another animation highlighting, for instance, the role of how the signal is being conducted.
Local current is strong enough to change the voltage around them, and that triggers their neighbors, which triggers the voltage around them, and so on down the line.
So let me just replay.
That local current is strong enough to change the voltage around them, and that triggers their neighbors, which triggers the voltage around them, and so on down the line.
So if you take a look at the entire picture, all this is just attributed to two major events. I may have the normal situation. Under depolarization, there is a flip. I have a transient negative outside, a transient positive inside due to ions moving in from the chamber. And then this eventually recover back to a positive and negative charge state. So this transient change appears to move like a wave. So we often think of it as a wave of depolarization, and hence being efficient to actually propagate the signal that we want. So I'm going to just show you another video.
Instead of just triggering one channel at a time in a chain reaction, a current can...
So if you take a look at these dynamics, it is very fast.
Instead of just triggering one channel at a time in a chain reaction, a current can...
So it is able to move between different nodes of Ranvier highly efficient. So this manner allows it to actually achieve a lossless conduction. So when we look at this as a form of neurocommunications, you may find that there are a lot of players that is important. As we sum up this talk, you'll find that neurons are critical, proteins are critical, particular channels, the maintenance of charges and its distribution is critical. And how does a cell achieve that? There is a reinforcing effect. And lastly, it is going to have some form of insulation to prevent the loss. So as we look at myelin, which is an important lipid, which is wrapped around the neuronal cell, particularly the Schwann cells, to prevent electrical loss. What it's trying to indicate here is if this insulation layer is thicker, as evidenced by the larger diameter, you will find that there is a better effect. So think of it, it's just like in a cold environment, you put on a jacket. If you go to a much more colder environment, you put on, for instance, a fur jacket, a coat, and so on. So the more you put on, the thicker the myelin sheath, the faster the conduction. So why is basically neurocommunication important? Because it is able to allow us to associate with diseases such as demyelination disorder. Think of it, patients that do not have the ability to put up sufficient myelination in their neurons, in their tissues, are unable to carry out neurocommunications efficiently. This may, for instance, affect the quality of life. So as we sum up the insights into neurocommunications, in this short talk, we want to highlight key importance of the molecular events, particularly the transient flip of the positive and negative charges along the membrane that allows the wave of depolarization to generate and accumulate in an action potential. And this action potential that is being driven progressively will be the key driving force to actually affect and to drive physiological events in the cell. So with that, thank you very much for attending this trial class. And this is part of the La Trobe experience that we're offering you. Thank you very much for your time. I will hand over the time back to Ming-Xue for a quick sum up.
So hi, about to. Right. Okay, hi. So good evening once again, everybody. So thank you, Dr. Seah, very much for your lecture as well. I hope that everybody here has actually gained some information about what's good about neurocommunications. So I think we'll just leave a couple of minutes. I'm not going to elaborate much, but I actually want to do a bit of Q&A if anybody has any. So if you do not, I think there's a chat function over there. Anybody has any questions for Dr. Seah or anything related to the program, you actually could click the chat function here and go ahead to ask him if you would like. Okay, we have a hand over.
Okay, so you can on your mic or to put in your contacts in the chat box so that we can respond to you. Lukman? Hi. So I have a question here. Is there ever a possibility that a neuron does not have sufficient potassium ions to trigger a nervous impulse? And is that a disease? Generally speaking, a neuron typically will have sufficient potassium because as part of the body system to conserve ions, our kidneys in particular has the high preference particularly to conserve potassium wherever needed. So before you excrete all these electrolytes via your urine as waste products, it will be fully conserved, and hence it is essential to maintain a basal level of potassium in all cells. In all cells, in fact, in your body. So for your next question, is that a disease? Typically, it won't be a disease, but typically it may be associated with physiological means of changes in potassium level, including a condition of low potassium level in the blood, termed as hypokalemia. Of course, hypokalemia for a long term may be going to be disastrous for the brain itself because it will then affect the quality of neurotransmission that is being conducted from the brain, which is simply means a key organizing center that may secrete, for instance, key hormones that regulate processes supporting homeostasis. So in short, typically cells will have sufficient potassium because it is preferentially conserved by the kidneys wherever there is a need to. But for a long term, low potassium will cause a key problem in affecting quality of neurocommunications within the body that is essential to maintain a body balance of all the events that's happening. Hope that answers your question. You're welcome.
All right, so maybe you can if there's any more last questions coming up from Dr. Seah, anything related to life sciences? Okay, I think oh, one more.
Okay, so the next one. Okay, so what happens if there is no absolute refractory period in a healthy cell? So technically, this is not possible because if that situation does happen, the only possibility is that the person have a genetic defect in the gene that encodes for the sodium gated channel. And should the person have such an effect, most likely it is embryonic lethal because you will most likely not be able to make it past, for instance, the birth, and you will not be able to carry out any cellular function. So should a gene defect in the voltage gated channels, particularly for sodium, being present, and hence there is not a possibility of no absolute refractory period being triggered. So most likely that particular mutation is going to cause a severe issue in neurotransmission quality for the patient, even if it is just going to be just a recessive effect, or one copy of the two copies is being affected.
All right, thank
Okay.
you. And okay, so any last questions? Okay, I think that being said as well, we also won't hold you back too much because we know it's 7:20 and some of you probably have not had dinner. We also won't hold you back to go and have dinner, but we'll just probably maybe just like to introduce a little bit about so what Dr. Seah is teaching is part of the La Trobe University curriculum, which I think he has mentioned, and it's a really brief insight about you if as you will choose to sign up for a program with La Trobe University as a student. This is what a blackboard online session will typically look like. And of course, not forgetting inside our STEM campus located in Toa Payoh, we also have our lab sessions ongoing as well for hands-on practical work. That being said, thank you everybody for really joining us in this session. So if any of you have any meetings related to program inquiries in terms of when's the next intake period, I think I've dropped you all an email about the link to sign up. I mean the link to sign up, join this session. So feel free to reply me accordingly, and then we'll link you up from there. So that being said, thank you, Dr. Seah, and thank you everybody for joining us tonight in this very insightful session. I hope everybody has gained some additional knowledge back for yourself and really helped you in your education journey. So that being said, thank you very much, everybody. Please have a good dinner and good night.